Thank you everyone for joining the webinar today on decoding in vitro oncology innovations, organoids, omics, and their role in early drug discovery and clinical application. Okay, so today I'll be discussing moving oncology models closer to the clinic to bridge the gap, specifically for in vitro systems, and how the use of innovations like organoid technology or high content imaging can be optimized into the in vitro assays, describing the key advantages of using either 2D or 3D in vitro systems, and when to incorporate multi-omics approaches into drug discovery programs. So we're all familiar with this typical drug development workflow, from target identification to lead discovery and lead optimization, which then moves into preclinical validation and eventually clinical trials. And this is a lengthy and costly process. So if we break down the early phases of lead discovery and preclinical development, you can see that there are many different in vitro, in vivo, and in silico tools and assays. And an early step in this process is screening of compound libraries for lead identification at an in vitro stages to reduce the number of compounds coming through to in vivo and clinical development. So drug screening assays often rely on high throughput screening approaches to gain meaningful insights within a reasonable time. So going from thousands of compounds to a few, ideally through this screening process. And this also depends on the type of therapeutic. So there's many different types of targets and therapeutics. There can be small molecules, there can be antibodies or vaccines, etc. So high throughput screening may not be applicable, but generally there are critical decisions made from those early stages of target validation, candidate selection, all the way through to IND filing for the identification of a suitable lead to take forward. So cell-based high throughput screens use cell lines which have been cultured and propagated in 2 dimensions. And this is in plastic, and these are very easily scalable and can also be used for downstream assays to interrogate the targets or further test the efficacy in lead discovery and optimization through to tumor-bearing models in vivo. And these models have formed the basis of our current understanding of cancer biology with extensive molecular and pharmacological characterization, which is very well published and they're widely available. But however, there was a steady rise in the use of patient-derived xenograft models in vivo over the last 10 years. And this has seen a shift in the use of PDX models instead of cell line models in the later stages to develop more translational studies. But there have been some limitations of using PDX or patient-derived systems in vitro. So there was a gap in the clinical relevance of these in vitro assays. So the need for more clinically relevant or patient-derived preclinical tools is driven by the fact that there's high attrition rates in oncology. And this report here shows the probability of overall success for drugs being approved, and this is based on obviously data in the last decade, at only 5% for oncology. And this is shown by the orange bar graphs and in comparison to the other indications. And the lowest success is occurring at phase 2 clinical efficacy stage. Now, despite this high risk of failure and the long and costly drug development process, the number of new oncology targets, the new agents, is still growing rapidly. And there's been significant advances also in immunotherapeutics, cellular therapies, ADCs and so on in the last 10 years. But there still is a lack of efficacy, especially in solid or high-grade tumours, off-target effects or toxicities that are not identified early enough, and also the lack of patient segmentation or the relevant biomarkers to identify the right patient segmentation. So we need better patient-relevant translational models to identify the right treatment for the right patient to improve those decisions early in the drug discovery process so that drugs are selected to progress forward. So if we look at how to improve some of the drug discovery process or workflow so that it can use more relevant models, so you can see that there are potentially 2 variables here that that need to be considered. The selection of a lead compound from a compound library typically in vitro screens, and then downstream in vivo modeling, and then the selection of indications among different disease types to identify the target patient population. Now, a drug discovery workflow to screen both variables at an early stage of drug discovery will provide great improvements in potential biomarker identification and eventually improve the agents that are entering the clinic. Now, there's been a lot of advances in mouse clinical trials using PDX models, which have been ideal for maybe screening some of this matrix approach, but it's impossible to do large screens with many different compounds or iterations. So you cannot readily perform a high throughput screening of compounds and models in vivo. So there really is a need to focus on the workflow for in vitro to incorporate patient-derived models if possible. So how can we do this? So as mentioned, there's many different types of therapeutics and different ways of screening them. But in addition, there's also many different types of models, some of which are patient-derived and some which are more patient-relevant. And that's just because sometimes patient-derived models are not always available for every disease type or indication. And I'll talk about this shortly. So this slide identifies the assay components that are needed, and then the assay format or the design are shown on the right-hand side. So firstly, one can look at how patient-relevant a model is that needs to be incorporated, and this could include the tumor heterogeneity, patient diversity, reconstituting the tumor microenvironment, and that can include many different cell types, and also the therapeutic target of interest and expression. And so the assay components will include different types of therapeutics and treatments, where the tumor source came from, and any other immune cells or effector cells that may be needed to be included in that assay design. And then you can look at potentially how high throughput that screen can be based on the complexity of the assay, the efficiency, the readouts or sampling, and then also the simplicity of the data analysis. So the assay design will have specific conditions like the number of wells, whether it's 2D or 3D, mono or co-cultures, and so forth. And the readouts will be dependent on the assay components and conditions. So it may not just be about tumor killing, it could be more complex. And then the downstream analysis, which will determine how the data is handled and processed. So you can imagine there could be different iterations of an assay design, depending on what components and design is used. And this is a bit like maybe having different ingredients in a recipe and building that to create a specific dish or an assay for a specific question or need. So this just helps to ensure that the in vitro screening system is built appropriately. And hopefully this also gives you an idea of how flexible and tailored assays can be. But this does require expertise in the different areas of 2D, 3D biology, drug screening, imaging, bioinformatics to help guide on building the right assays and analysis to ensure the right output is achieved. So today I'm going to cover some of those tumor sources, conditions and readouts and analysis, and give some examples of the different types of patient relevant assays that can be created. So let's look closely at some of the models that we can select and the design options. As mentioned, the poor attrition rates in oncology really does highlight the need for better translational models for both in vitro and in vivo. And these models have come under scrutiny, especially the cell line models, for failing to better predict clinical efficacy. Now, cancer cell lines are grown in two-dimensional culture, and they have been the workhorse for many decades. But the relevance of these models has been debated, and so collections like the NCI-60 panel, which is shown here in this paper, have been retired in favour of patient-derived models that haven't adapted to grow in plastic or 2D. So the patient-derived systems are seen as gold standard, and they potentially can bridge that gap between the clinic and the patient. So the lab and the clinic, better than some of the traditional models, because they do actually preserve the original tumor histopathology, the molecular pathology. They recapitulate the inter and intra-plate patient heterogeneity, and also represent patient diversity much better, especially if there's large collections of models being developed, which can actually give better indication of clinical efficacy. And they have also been shown to be pharmacologically predictive. So these patient-derived systems include PDX models, primary cells, patient tissue, and organoids. And PDX, for example, have been widely used for translational studies, and they've been shown to be highly predictive of the clinic, as shown by this paper here. But the inclusion of these systems in early discovery has been limiting, and as such, and this is because they are actually maintained in vivo. Now, this is where organoid technology has had a significant impact. So like PDX models, organoids are also created from patient tumors. You can create biobanks, you can capture that patient diversity and heterogeneity, and you can also do clinically relevant studies. Now, multiple studies have reported the clinical predictivity of organoids, and this paper here was one of the first ones to show the clinical relevance of organoids, using them in testing, for example, in metastatic colorectal cancer, which were derived from patients. So using a compound library of drugs, the group were able to test multiple organoids, looking at the sensitivities versus the actual patient response in the clinic. And they showed 100% match in the negative responses and 88% for positive responses. And at Crown, we've also developed platforms with patient-derived models, which include in vitro organoids, in vivo PDX, as well as ex vivo patient tissue. And also we have primary cell lines. And this covers many different cancer types and can support drug development and translational research. But we do also have a large bank of cancer cell lines. So we have over 700 in our collection. And then we also have over 200 models, in vivo xenograft models, to match these cell lines. And these can also be used at different stages in the drug discovery process as well. So they are still valuable and still useful for research. So today I'm going to focus just on the in vitro, and I'll spend a bit of time defining those organoids and the 3D assays in a bit more depth, their applications and their uses, together with the 2D cell lines, and also applying those downstream omics tools. But we do need to take a step back a bit and look at what we mean by patient relevant, and also recapitulating that tumor microenvironment in vitro, because it's not the same as carrying these studies out in vivo. So there are different types of in vitro cell-based assays, especially in how the 3D cultures are constructed. And this is represented in this figure here. So you have your standard 2D assays with the cells cultured in flat monolayer using homogeneous cell population. This is shown on the left-hand side. And you can even use cells from the primary tissue if there is a stable culture that can be achieved. And as you move through into more complex 3D format, the cells or the tissue can gain more relevant cell-to-cell interactions and cell matrix connections mediated by integrins. They form spheroid structures when embedded in support matrix, such as matrigel or collagen. And there's different types of 3D systems as well. So these can be either anchorage independent or scaffold-based 3D systems. So this adhesion and cytoskeletal organization and cellular signaling becomes more true to those in vivo situation, which then impacts how cells proliferate, their differentiation, their migration and survival. And so as you can see, as you go from left to right, you start getting more complex. So for example, especially when you start including things like patient material, these will then become more patient relevant. Now you can also admix in other cell types. So this can be stromal cells, such as fibroblasts, immune cells, and again, this makes these more complex. And as you get closer to, therefore, constructing the tumor microenvironment in a more relevant fashion, the assay is more complex, and therefore the throughput can potentially decrease. So these don't necessarily mean that these will be high throughput. You can still use them for screening, but there is a limitation. And also the readouts can become more complicated with these multicellular co-cultures. So the type of analysis needs to be optimized. Now, to simplify this, I'm just going to focus on referring to them as either 2D, 3D organoids for this webinar. So some of the key practical features are summarized here. So the 2D cultures, which, as I mentioned, are very widely used as they're easy to set up. They're derived from monolayer cultures, and their application is actually very versatile. They are flat, with 50% of the surface area being in contact with plastic, and they're typically a single cell population. But the advantage is that you can actually expand, and you can manipulate and cryopreserve and bank these models. In 3D, the single cell populations, or even mixture of different cells, can be used, including primary cells or even human tissue. And these tend to form spheroids or cell clusters. And spheroids can mimic different proliferative and quiescent phenotypes, hypoxia, as well as you can look at drug penetration and resistance. And therefore, they potentially can predict better drug responses when compared to the same cells grown in 2D. However, unlike 2D cells, they cannot be maintained in long-term culture, and in some cases can undergo senescence, especially when fresh tissue is being used. Now, spheroids are very different to organoids, and sometimes the two terms are incorrectly used or interchangeably used. So it's important to understand the differences between these models and how best to use them based on the properties. Now, organoids can be developed from either pluripotent stem cells or adult stem cells. So the one that I'll be referring to for this webinar is derived from adult stem cells. And this is using the technology developed by the Hans Cleaver Lab. And the key difference is that organoids have self-organizing properties that are made up of a mixture of stem cells and differentiated cells, and you can find, just like you can find in those sort of corresponding organ that they've been derived from, or the tumor. And the structures resemble the sort of microanatomy of the tissue from which they originated, which is a key differentiator in comparison to the less complex spheroids or the 2D cultures. In addition, the organoids have the ability to be maintained in long-term culture, just like the 2D cells. They can be passaged, expanded, and they also can be backed down. And that is because of those stem cells, which can replenish the tumor material, the tissue. And this cannot be achieved, obviously, with the spheroids. So the term organoid sometimes is used to describe mini organs or tumors derived from those stem cells. So the first question is then, why use 2D cell-based models? So I'm just going to, so this slide just summarizes some of those key features as well as the advantages. And because of these key features and those advantages, you can see how flexible and easy it is to use 2D cell lines, and therefore they become the sort of go-to model right from the outset. But because of the limitations, there has to be some caution in what they're being used for. So the applications where 2D cultures have the most impact is for things like biological investigations and more and potentially non-translational studies. Definitely high throughput screening for lead identification, also lead optimization. They can be used in target engagement, looking at the oncogenic pathways or mechanism of action. They can be easily manipulated by engineering, so they can express specific mutations or knockout. And they also can be used to develop more complex in vivo models through some of this engineering. They also can be used to generate hypotheses like biomarker identification, but this will then need to be qualified in more translatable systems. And because of those advantages, the cell lines provide and again, this includes the primary cell lines in this, you can actually do large scale cell panel screening. And we've actually created this platform which can be quickly used to evaluate different drugs in a large number of cell lines at once to retrieve responder, non-responder profiles. And we call this OmniScreen, which is run on a quarterly basis. So you can register for it, sign up for it, and the screen is run and the data is then provided in a couple of weeks' time. Now, this is very high throughput. There's at least 50 different cell lines that are run in each quarter. The cells are of high quality, so they've been SDR profiled, they're mycoplasma free. There's positive drug controls that are included. The cell revival is all ongoing. As well as the seeding densities across different time points is already being optimized. So this becomes very cost effective. And you can focus your screening panel based on cancer type, on the mutation of interest, or even the availability of matched in vivo models for downstream studies. There is also bioinformatics support throughout the study to optimize study design, as well as the data analysis. And then you can review your data in real time through our online sort of client portal. So by combining the efficacy data with the genomic information of the cell lines, we're able to then identify novel targets and also hypothesize on the drug mechanism of action, as well as providing predictive biomarkers. So I'll give you an example a little bit later. So if we look at the 3D system, so what is the best application for these types of models based on those advantages as listed here, but also remembering the sort of limitations. So again, a wide range of different cells, tissue, co-cultures can be used here. They do closely mimic the tissue architecture with those cell-to-cell interactions. So they are more physiologically relevant than the 2D systems. But because of their limitation of being maintained in long-term culture, it does impact the sort of reproducibility and the robustness of the assays, and you do have to generate each of the systems each time. Now, so the applications, again, they can provide insights into the target engagement, the oncogenic pathways, the mechanism of action, efficacy in a relevant tumor microenvironment, and that's the key difference here. They also can be used to model cancer stem cells, hypoxia, drug penetration studies because of that 3D kind of structure, and also look at resistant mechanisms. So a lot of clinical hypothesis testing can be done, especially when you're using tissue from patients. So for organoids, hopefully you can see that there are many advantages over the other systems because of they are clinically predictive, derived directly from the patient material. So they preserve the key features of the originating tissue. And like 2D cell lines, they're very scalable and they can be biobanked and used over and over again. And because of the fact that they're reproducible and they're easy to use, they can be automated and used in high throughput screening. Now, some of the limitations are that it's actually it's not as easy and efficient as the 2D models because there is a little bit more complexity in actually culturing organoids. So the throughput may not be as high. But the applications, because of that throughput, is still sufficient to do a matrix high throughput screen. And they are sometimes referred to as clinical trials in a dish. You've got many different models that can be used across different patients, so you can actually capture the diversity. And you can also then have matched in vivo downstream models to progress if needed. And then also these can be applied to different stages of drug discovery. So you can do studies as early as target validation, all the way through to biomarker identification, as well as engineer them and do co-culture studies. So there's potential using for tox studies using the normal tissue, as well as immuno-oncology studies because of that co-culture capability. So this slide just summarizes the 3D in vitro models. So the spheroids and the organoids, which better replicate human biology in vitro. And they're also, as mentioned, amenable for a range of different applications. So patient tissue or even PDX tissue, as well as cell lines, can be used in those 3D spheroid assays for rapid screening in vitro, whereas patient tissue and PDX tissue can be used for organoid development, which is shown by this schematic in the middle, which defines or shows how the PDXs from the the organoids from the PDXs are created, which are called PDXOs, and then also the organoids directly from the patient, which are referred to as PDOs, and then their eventual xenograft generation. So the tissue specific adult stem cells within these tumors allows the organoid generation from both patient normal tissue, but also from the tumor material. So this means that you can actually create a number of different models, matched models. Using our vast sort of collection of PDX models, it means that you can actually increase the number of tumor models in your collection as well. So we currently have quite a large collection of over 400 models that are available in our database, which are the PDXOs, and over 200 models that are PDOs. And like I said, there are matched models for the downstream in vivo, as well as normal normal matched organoids for some of the tissue that are shown here. Now, generating those biobanks of large numbers of tumours is really important here, because that will then capture that patient diversity for drug discovery and screening. So in this paper on the left-hand side, we actually describe the methodology for creating the organoids from our PDX models, and then biobanking and characterizing these. Now, the self-renewal capability of stem cells means that you can indefinitely expand organoids in long-term culture, but still maintain that genomic stability, which again is shown by the graph on the bottom left. You can also generate sufficient material in a dish. So again, you can do large scale screens. And then you can carry these studies on afterwards, because they can be replenished and revived from cryopreservation. So the paper also describes the application for drug screening, looking at the robustness and also the how the matched models are able to predict the in vivo response. So there's very high correlation between the in vitro and the in vivo. And then the review paper on the bottom right also reviews how within the field organoids and the PDXs are being employed for different cancer types across different therapeutic modalities. And then also there's a webinar which is shown in the top right hand corner, just that you can learn a little bit more around these matched models. And then this slide just gives you a breakdown of the different cancer types. So we have over 15 different tissue types in our collection of organoids. Again, this can be found in our online database. And then similar to the PDX biobank, our organoid biobank also has fully annotated information for clinical information, pathology, diagnosis, genomics. And you can use a database to actually search the whole collection and pick the models that are of interest. At AACR this year, actually, we presented a poster on how we are increasing the throughput of organoid screening by forming assay-ready organoid drug screening platform. And this covers different cancer types, and there will be a set number of organoid models. So very similar to that OMNI-screen I described earlier. This will be available for registration on a quarterly basis, so it can be more cost-saving as all the optimization and planning has already been scheduled. So look out for screening opportunities in the near future using this platform. So one of the assay design considerations for any biological or complex 3D assay, as well as co-culture, is the incorporation of high content imaging. And this really allows capturing a lot of information within these complex cultures. So the sort of temporal spatial analysis at the cellular level, you can look at the size and the shape of the cells. It's not just about tumor killing. And this also complements high throughput screening. So it creates a vast amount of data. It's fully automated and can produce reliable and reproducible data. It can reconstruct the 3D images to visualize the effects and also provide quantitative phenotypic readouts so that you have additional information as well as the tumor kill. The high content imaging and the data is then analyzed in-house by our team with the proprietary software. And this has been specifically developed to measure those different sort of phenotypic changes. So this leads to the next section of the talk, which is looking at the sort of power of analysis and the critical steps for biomarker discovery. And there's many different tools that you can use downstream for analysis, which can be applied to in vitro assays, as well as the sort of large scale drug or model screens, which I've just been describing. And these services are shown here. And there's, like I said, a different range that can be applied for different assays. And these lab services can also be used to create baseline data for the models as well, not just for ongoing sort of studies. So this sort of baseline data can be found in our online databases, which I've kind of touched upon as we've been going through. And these are so you can register online, you can search through a wide range of tumor models. So we cover PDXs, cell line models, xenografts, organoids. There is, the data includes pathology, patient details, next generation sequencing, proteomics of certain models, histology and pharmacology. And so for in vitro organoids, you would be looking at organoid base, and then for the cell lines, you would be using xenobase. So take a look at these. This is really useful information to give you a bit of background as to those specific models. So the baseline data that is in these databases for these different models can be used to used in the screens and then eventually be used for biomarker discovery once we know what the responder non-responder profiles are like. And studies have shown that clinical trials that use biomarker strategy for patient selection are often more successful when compared to those agents that don't have an associated biomarker. And also actually identifying those biomarkers early in the drug development process can actually help produce a robust biomarker guided approach to identify the most promising therapeutics, as well as the right patient populations and indications. So specifically for in vitro screening, which can be very fast and there's going to be a lot of data created, the biomarker discovery here is aimed to a different sort of parameters. So you could look to explore or validate drug mechanism of action. You can identify those genetic features that are associated with drug response. You can develop either single gene or even composite biomarkers, and also use the data for guidance on future downstream model selection. So for this last part of the webinar, I'll give you some examples of different types of assays that incorporate different components and different conditions for these specific therapeutics. And as mentioned earlier, the design can be tailored to meet very specific needs based on the therapeutic agent or the stage of development. So using this format again, so the first study that we'll be looking at is one that involves irinotecan, and this agent interrupts the cell cycle in the S phase, causing cell death due to the DNA replication machinery being halted. So the aim here was to develop a composite gene signature biomarker that could potentially be used for clinical use. So the design included, so irinotecan efficacy was assessed across different cell lines. So we're using over 250 different tumor cell lines. And this covers different indications in a simple 2D assay using responder, non-responder profiles and the cancer cell line encyclopedia genomic data to identify those potential biomarkers. So here's the plot showing the distribution of the 256 cell lines response, which is represented by the area under the curve and separated out by tissue of origin. And we observed significant differences across the solid cancer types. The large sort of volume of efficacy and potency data obtained from this sort of in vitro screen was also used to classify the cell lines in terms of their irinotecan sensitivity. So there's three groups: the insensitive models, the sensitive ones, and then the ones in between. So about 86 of the models were shown to have low sensitivity, and 86 were very high. And there's obviously an in-between, which is not quite clear whether they respond or not. Now, next, we did the global gene expression analysis of all the cell lines and assessed this by the principal component analysis, which is shown on the right-hand side. This is to potentially identify maybe a natural clustering of the groups shown within this these cell lines. But we found that the transcriptomic analysis on these solid tumor cell lines showed no obvious clustering based on the sensitivity or non-sensitive profiles using 20,000 sort of human gene expression data. So we really do need to look for a more specific molecular marker or biomarker. So next, the functional analysis by gene ontology of biological processes identified DNA replication as having the most differential gene expression between responders and non-responders. And this was regardless of the number of top genes selected. So this is shown in the teal graph here, and these were always enriched for DNA replication. This result obviously is very consistent with the proposed or the known irinotecan mechanism of action, and also highlights that the value of using cell lines in vitro screens, as they still can give relevant information. But what was interesting was that SLFN11 has also been identified or has been reported as a single gene expression biomarker for irinotecan sensitivity. Now, this is a putative DNA RNA helicase that has been shown to be shown to irreversibly block replication in cells that are experiencing that replication stress. So cells that express high levels of SLFN11 have been shown to be more susceptible to DNA targeting drugs in specific cancer types like breast cancer. But it must be noted, typically, or taken into consideration, that single biomarker strategy is not always possible for every therapeutic, and it may, in some cases, those single biomarkers may not ideally reflect the outcomes, or the complexity of many other diseases, such as cancer. Some caution should be taken with the single gene approach. And also, in an era or an age where advances in technology have opened the door to being able to measure multiple parameters simultaneously, this is now leading forward the construction of composite biomarkers that provide a more complete picture of the biological system. So here in this study, we took the data, and the highest prediction accuracy of irinotecan sensitivity we could see amongst a set of 21 genes, which are shown here. Now, the composite biomarker panels can be complex, so it's key to actually make sure that there is further downstream validation steps taken once that sort of gene signature has been identified to make sure that there is reproducible or there's accuracy and sensitivity in further tests. So doing this once is not enough. You have to reiterate it. So the 21 gene panel here was also evaluated on an independent validation set to report out the sort of positive and negative prediction values. And as shown here, the correlation between the area under the curve and the signature score in all the cell lines, including those medium sensitive group, was clearly higher with the composite biomarker compared to the single gene biomarker. Now, so while the expression of SLFN11 was the strongest single gene biomarker, the expression-based 21 gene composite biomarker was able to achieve a higher efficacy prediction. This could then be further, so this can be taken further and qualified in more patient-derived systems. In the second example, the objective of this study was to identify drug resistance using patient-derived models in 3D. So this included small molecules or targeted agents such as HDAC inhibitors, chemotherapeutics, or standard care doublet treatments that are typically first and second line therapies for non-small cell lung cancer. And again, this was to see which models or patients were resistant to those types of treatments. So PDX material was used here. It can be dissociated, and it was taken from 30 different models, put into these 3D ex vivo cultures. And these were also some, in cases, co-culture with stroma cells, such as cancer-associated fibroblasts or bone marrow-derived mesenchymal stem cells, which are known to support resistance. The assay was miniaturized into a 384-well format. So it was a 3D complex co-culture system supplemented with Coltrex, which is a laminin-based membrane extract to support the growth of the PDX tissue. And it was simply just to look at tumor killing and then determine the responder, non-responder profiles and potential mechanisms of resistance. So this diagram shows the typical cycle for PDX screening in vivo, where we're generating the PDX from a primary tissue is initially done and then banked down to create a biobank, and then responder profiles are evaluated in vivo to generate those gene signatures and biomarkers. And this is quite a long process when it's done in animal studies. So being able to take the PDX material into 3D ex vivo culture is much faster, which is shown by the sort of skip through the middle of the diagram. Actually, this work was published, which is shown on the right, and the difference in response in the models is shown as a heat map. So you've got the different sort of conditions across the top and the different non-small cell lung cancer models across the side. And we reported that in some cases there was increased resistance in the presence of those stromal cells, and this was then further qualified with in vivo studies. So this assay is very versatile, as you could admix in different components, and the readout was very simple. But actually assessing 30 different models using fresh material from the animals was not as efficient as using organoids. But this is still a valuable tool, because in some cases organoids are not available for every single patient-derived model. So therefore this is sort of a halfway house in making sure that we can still use those models for in vitro screening. And sometimes in some cases there may be some types of organoids that are organoids that cannot be derived from non-epithelial based tumours. So again, these types of tumours can be included into these ex vivo assays. So the next example actually is a very good demonstration of how fast and efficient organoid screening can be, and also that you can include antibody screening into these study designs. So this was using treatment with a range of different biospecifics in colorectal cancer organoids in a high throughput manner in multiple time points to assess phenotypic response via high content imaging, and also potentially identification of biomarker response. And again, this study was published in Nature Cancer last year, and we were able to demonstrate the value of high content imaging with organoids for an unbiased functional testing, which ultimately led to a lead agent or lead candidate moving into clinical trials. Now, initially, several organoids models were used in the primary screen, so the first screen, and this was looking at over 500 therapeutic antibody candidates to narrow down a selection to a handful, so that these could be then used in a second screen, which used over 20 colorectal cancer organoid models to really kind of define the differences in response across a larger cohort of patients representing different representing patient diversity. And to select in the end a compound that showed very high selectivity towards tumor organoids. But also, because normal organoids could be included, we were also able to assess the response to the lead agent in normal situation, as well as in comparison to benchmark agents such as cetuximab. So the data here on the right shows the internalization and degradation of EGFR by the test agent, which was actually more rapid in comparison to cetuximab. Also we found that it was mediated by LGR5, which is expressed in the colorectal cancers, but not in the normal organoids which lack LGR5. So this led to the identification of a single agent targeting EGFR or LGR5 positive cancer cells, cancer organoids, with limited toxicity to normal tissue. And this drug, as mentioned, is now in clinical trials. In another organoid study, the synergy of drug combinations was assessed. And this was quite a simple study based on small molecules, a couple of organoids, but in a high throughput way, so that we could actually do different combinations and just simply looking at tumor killing. So the agents being tested were a WEE1 inhibitor and a CHK inhibitor, together with a standard care agent, gemcitabine, in pancreatic cancer. Now, there's evidence to show that these agents have synergies in p53 mutated pancreatic cancer, as well as other cancers. So we decided to test this hypothesis using pancreatic organoids with p53 mutations, similar to the published studies. And we used our database to identify these models and selected these models based on those p53 mutations. Some were nonsense mutations. And the first, we looked at the sort of single agent response, just so that we could assess the sort of dose range of sensitivity for those individual agents. And then we could then design the next stage, which was the combination analysis. So this published study, which describes this assay, involves treating those organoids with a 6-point titration of each of those inhibitors. So it's a 6x6 matrix combination design. The treatment is on for 5 days, and cell viability is measured at the end. And to analyze the data in a pharmacologically meaningful way, 2 mathematical models of synergy were used to assess that combination effect. So the first one is the Bliss independence model, which is expected to hold true for non-interacting drugs, which elicit a response independently. So this is looking at two different pathways. And it looks at the power of those two compounds together over monotherapy. The allowed additivity model is more compatible with cases where both drugs have a similar mode of action, for example, on the same target or the same pathway. So it looks at the power of the two compounds together than alone. And generally, when we are looking at the synergy score, if it's higher than 5, it indicates good synergy, but if it's lower than 5, it indicates poor synergy or even antagonism. So, we obtained the combination data for the 3 different compounds in combination with each other across 3 different models using the 2 mathematical models. So again, there was a lot of data, but here I've just shown a selection of the data to demonstrate the readout and the analysis that was conducted. So, for example, the combination of those 2 inhibitors shown here in one of the pancreatic models. So on the left-hand side is a dose inhibition heat map for the 6x6 matrix combination with the percentage inhibition. And the increasing intensity of red is obviously the increased association of inhibition. So there's a total of 49 data points, and this includes the zeros as well. Now, in the middle, the synergy score that has been created by the 2 different mathematical models is shown in that middle diagram for where the similarities between the methods can be observed, and the red shows the synergy intensity, and then the low or antagonistic score is shown in the green. The synergy score can also be represented as either that 2D contour map or also a 3D response surface plot, which is shown on the right. And generally, the combination of those 2 inhibitors showed very strong synergistic effect in this model, with very high scores above 10, in some cases 50, which is shown by that intensity of red. And actually what we found was that the highest synergy score was seen at lower concentrations, which is shown by the arrows on those diagrams. Whereas some of the higher concentrations actually resulted in antagonism. So this really kind of highlights how the synergy scores can be shown visually, and how you can pull out the different types of data based on the complexity of the interaction of the two drugs. And so here I'm just showing you the synergy score heat maps for the combination with gemcitabine across the 3 models, focusing on the areas where the synergy was high. So this is highlighted with the sort of boxes. And what this demonstrates was that the high synergy scores were found to be at lower drug concentrations, again in 2 of the models, which suggests submaximal dosing to be preferable for those specific patients, especially when you're considering that combination with gemcitabine. Now, in comparison for one of the patients, PA1252, the peak quadrant was actually higher, so the submaximal dosing would not have been beneficial for this patient. And obviously, in one of the models, in PA2487, one of the combination was significantly stronger than the other. And again, this just shows that this type of combination will be beneficial for some patients, but not for others. And overall, the differences in synergy score was observed across, the p53 mutated models, depending on their dose combinations, which again correlates well with some of the clinical studies that have been conducted. And this also highlights again how organoids can be really valuable in trying to understand how to use different drugs to treat the right patient. And this final example, so this one is slightly different in that we're now looking at assays for investigating immunotherapeutics, and the design of the assay will incorporate some additional factors that reflect the tumor microenvironment. So here we can look at both immune checkpoint inhibitors and small molecules in patient tissue that has been dissociated, incorporated into an ex vivo assay. And as the tissue is directly from the patient, it can still retain many of the different effector immune cells or stromal cells, and these are referred to as being endogenous. And the assay does require supplementation in form of a hydrogel matrix, and the throughput is dependent on the amount of viable tissue and the availability of that tissue. So as this is a complex assay, the tissue structure and the readouts need to be measured. So it needs to incorporate high content imaging, as well as some of the downstream analysis. You can actually incorporate other readouts such as cytokine analysis or flow cytometry, next generation sequencing for these types of assays, just to get kind of more information, because these are obviously very valuable tissues. This ex vivo patient tissue platform is actually one of the most patient relevant and models available for screening drugs, as it uses fresh patient tumor. And this tumor is processed within 24 hours, so it preserves that native tumor microenvironment and those endogenous cells. There is also a webinar that further details this process, but just to show that this is actually a very quick assay, as there's no culture or establishment in vitro needed beforehand. It can be high throughput. Drugs such as chemotherapeutics, standard of care agents, immunotherapeutics can all be used. They're assayed for between 5 to 7 days. And then the wells are fixed and imaged, and the phenotypic readouts will be analysed, giving information more than just the sort of sperm cell viability. Now here at the bottom, the panel of ovarian organoid models have been ranked to their drug sensitivity and resistance to carboplatin, and the heat map visualises the sort of patient tumor response to other agents like paclitaxel, targeted agents like PARP inhibitors. You can also pull out models with specific features like BRCA mutations versus wild type to see which patients are responsive or resistance or show resistance. Now, you can also use this assay to evaluate those immunotherapeutics because of that intact native immune cells. So here, the same example of ovarian tissue, the sensitivity and resistance has been ranked against the response to a super antigen. And if you pull out patient 27 here, which is a BRCA mutation, you can see that there are differential responses to different checkpoint inhibitors. So this patient, for example, was very responsive to ipilimumab, but not to nivolumab or pembro. However, this can be very much dependent on the tumour and how hot or cold it is, and the level of immune cell infiltration at the time of that tissue collection. So here, the infiltration of CD3 positive T cells, as well as other markers like Ki67 proliferation, or markers for ovarian cancer, can also be assessed by IHC, just to kind of get some more information about why the tumours may be responding to those immunotherapeutics. So this assay is actually very versatile, and obviously the limitation is on that tissue sourcing and the amount. So for example, for triple negative breast cancer, it's very easy to get tissue, whereas other subtypes it may be less so. Okay, so to summarize today's webinar, the key takeaways are, and hopefully you've seen that patient relevant models can potentially bridge the gap between the lab and the clinic, and reduce the attrition rate in oncology. And actually, by front loading the early in vitro stages of drug discovery with more patient relevant biological models, this will certainly increase the quality of molecules entering the clinic. The advantages and limitations of the different 2D, 3D systems, as well as the different sort of innovative technology and downstream analysis, also need to be understood so that the in vitro screens can be optimized. Introducing biomarker structures early in preclinical studies and also has many advantages, but critical is having the expertise in designing the right screening assay using those different 2D, 3D systems or patient-derived models, as well as multi-omics approaches, so that the right drug enters the clinic for the right patient. So with that, I'd like to thank you for your attention, and I'd be happy to take any questions.